Hi everyone. In this session, we will talk about PCA, the principal component analysis, and it is one of the primary component, I would say, or one of the main component of the single cell data analysis. Okay, that's what we will focus on and why we need to do PCA. Okay, that's a very important thing that we will discuss. So in any single cell data analysis step, so these are the steps usually followed. And already we have covered these 4 steps in my previous videos. Like you start with the raw data and go with the filtering. So where you remove the noisy cells. Then once you have a clean data, then you need to do normalization. That's where you are removing the technical noise between each cell. So now cells are now comparable after normalization. Next step, you go for scaling. This is where you are focusing on the genes. So that means you are making sure that the expression of the genes are now comparable. So now after these 3 steps, our data is now normalized and scaled. That means we have removed the noise between the cells as well as the genes. So next, instead of focusing on all the 20,000 genes, because which will be very huge, we now focus only on the top variable genes, because those genes which are not variable, they are not important for clustering. So that's why we are not interested in them, so we ignore them. So usually 2000 genes are very usually enough for single cell data. So that's where we find the top variable genes. So now the next step is the PCA. So basically in the next step we run PCA with these 2000 genes what have the signal, because those are the variable genes that we have identified. And then we actually, PCA is a very compression kind of technique. Okay, for example, how we zip your files, that will reduce the file size. Same way PCA will also do some compression method. So instead of now working with 2000 genes, you can say that we will work with just 50 metagenes. Okay, so each principal component is kind of one metagene, you can say. We'll talk about that, what PCA is all about. So basically in this step, instead of analyzing 2000 genes and let's say 2000 cells, we will compress that into 50 metagenes and with the 2000 cells. Okay, so what is PCA? PCA is the principal component analysis, right? So basically it is more of kind of, you can say that it's a data compression method. So basically in your data, you have the signal and you have the noise. So basically you are removing the noise and taking the signal. Okay, so that's what PCA does. Okay, so if you take an example, let's say this is our actual data what we get, where we have each rows we have genes and each columns, let's say, cell. In this hypothetical example, let's say we have 20,000 genes and each column represents cell, and we have, let's say, 2000 cells. So now you can imagine the size of this matrix, 20,000 by 2000, right? And each value is a matrix. So this way, this is a very highly redundant and noisy data. So this is genes by cells matrix. So what you do, PCA is like you take this data and somehow compress it. I will tell how to compress that, but let's say you have some compression method. So now using that, now you can get another similar matrix kind of structure. So now instead of genes now, so we have principal component. So we have PC1, PC2, PC20, even PC20,000. So basically if your input is 20,000, we will get 20,000 principal components. Okay, and the number of columns will be remain same cell. So basically your input space is the actual gene, whereas the out after PCA, we call this a PCA space. But only difference between these two is like, so here the information is scattered across all the 20,000 genes, but here the information, same information is captured in the top, let's say 10, 20, 30 principal component. That much information will be stored. So now instead of taking all these 20,000 rows, you can just pick up these 20 rows, for in this case let's say 20 principal component, and that much information, both will be same. So either you take the raw data 20,000 by 2000, or you just take this 20 by 2000, that will contain the similar information. So from 21 to remaining 20,000, those are the noise, so we can simply ignore. So this is how we do that. So your input space was 20,000 by 2000, your output space was again 20,000 by 2000 only, but only difference is like in this 20,000 you just pick up this 20, that is enough to when you say that you want to take 20,000. So this way now your 20,000 by 2000 is now reduced to just 20 by 2000. So this is how the compression works. So how does it work actually? So now if you take this PC1, right, the principal component 1, which contains the maximum variance in the data, or maximum information it stores. So what is PC1? PC1 is nothing but, you can say, some weightage from the gene 1. So you can say that gene 1 contributes to some percentage. gene 2 some percentage, gene 3 some percentage, and like that 20,000 genes. Each gene will have some proportion of the information passed to the PC1. Similarly, some again for PC2, some other genes will be contributing towards PC2. PC3, some other genes will be contributing. So now PC1 is derived by as a linear combinations of the information from each genes. That's why the information what is there in this all this 20,000 gene is actually stored in the PC1, PC2, PC3, PC20. Okay, but the only thing also now important things to know that which genes the information of which genes are passing to which PC component. That's very important. So that's where in PC analysis, once we compress, we need to know in PC. So now for you PC1 is nothing, you cannot understand what is PC1, right? Because you understand gene, biology is the gene. PC1 is simply mathematical X information. So you should also need to know when I say PC1, actually what does it mean. So in that case, what you do, you come back here, you look at this formula and calculate, hey, oh, PC1 is nothing but, let's say in this case, gene 2 is contributing more because it is giving 0.15 compared to gene 1 or even compared to gene 3. So in this case, you say that, okay, gene 1 and gene 2 are passing more information to PC1. That means these two are correlated with the PC1. So that's what we need to, we check in PC analysis. So if I just summarize what I did, like you take the input space, you convert that into PCA space, and then you just find which genes are correlated with which PC component. So that way we need to, that's what PCA does. Okay, so now after PCA, we don't take this one, we instead take, sorry for that, we take this 20 cross this one data matrix. I hope clear. So let me further explain like PCA what it does when I say how it calculates, how it compresses, that I will show you. So in this case, let's say there are two axes, okay, two dimension. Okay, this is the original, let's say gene 1, and here gene 2, and each dot is equal to, let's say, one cell. And let's say this is the first cell. Here the gene 1 expression is, let's say, this one, whereas in the second cell the gene expression is like that. Okay, and similarly this cell like that. And for example, when you take this cell, in this cell, your gene 1 expression is, let's say, high, and gene 2 expression is like that. So each dot is equal to one cell. So now if you see, if you take this coordinate original gene 1 and gene 2, basically your it goes like that. That means it is going along the diagonal, right? That means in order to explain this expression value, you need both x and y. But what PCA does, like let me just sorry for that. What PCA does is okay, sorry, I could not pause it. What PCA does is PCA stops here. So basically this is the direction where your maximum variance is there. Okay, so basically now instead of, so this PCA1 will be like this. So that is the direction where your maximum information is there. Okay, and PC2 will be perpendicular to that. So that's the orthogonal we call. So PC1, PC2, PC3, PC4, these are all perpendicular to each other. That's why it is very, sometimes we call it linear. Okay, so PCA is nothing but linear dimensionality reduction. So in next videos downstream of the single cell analysis, you will see UMAP and TSNE. Those are non-linear. Okay, PCA is linear, where those are non-linear. I will talk about the difference between those, but you get an idea. Like PCA is performs a linear transformation. It assumes that the most important information in your data can be captured by drawing a straight line through the multidimensional space. So that's why linear line is a straight line, right? So that's how it derives the information. So that's why PC1 is equal to some weightage of gene 1 plus some weightage of gene 2 plus some information from gene 3, like that it calculates, and that's why it's a weighted linear combination of the original genes. So that's why we need to know which gene is contributing to which species, positively and negatively. Okay, so that's why because of this straight line projections, right, the PCA preserves the global distance. Okay, so that's why what it means, like if two cells, okay, are far apart in the original 20,000 gene space, they will also be far apart in PCA. So for example, in the principal space, if two dots are very far, means indeed their expressions are very different. So that's why we are taking PCA as the foundation for downstream analysis. So you cannot take, you cannot do UMAP or TSNE without PCA, because PCA will remove the noise and capture the signal, and also it captures the global distance, because in the PCA space the distance between two dot or two cell is represents the actually in the original space, in the gene space, how far the expressions are. Okay. Okay, so if I come back to this, like practical, right? So far what we see, like as I said, in the single cell data analysis, this is the guided tutorial that we have been following. So you load the data, then you do the standard pre-processing. That means you do the QC and choose particular cells based on this number of features or number of counts or mitochondrial percentage. You can do some filtering. Then here we are doing the filtering here. Then you are normalizing your data. Okay, and then you are finding the highly variable genes. Okay, and using that you are doing the scaling. Scaling means now you are doing the all genes into a same level, gene expression of the genes, and now we're doing the linear dimensional reduction. And for that there is a function called RunPCA, and here we calculate that pass that object and how many genes to take. As I said, right, you cannot take 20,000 genes, because in the various previous step we have identified of the 20,000 genes only 2,000 are the most variable genes. So that's why we can take those 2,000 genes where the signal is there to do the downstream analysis. Okay, so RunPCA is the function that it does. So it has some other parameter that we will explore. So now let's jump to the practical here. So I have already done the previous steps, but I can quickly show as well. So you can load the library, load the data, then you create the Seurat object, then you calculate the mitochondrial percentage, and these are the plots. And then basically we need to filter. This is the filtering that we are doing here, and this is the normalization where cells are this thing. Now we are doing that, finding the top 2,000 variable genes, and yeah, and here we are doing the scaling. Okay, so after the scaling you will do the run PCA method. Okay, so here we are taking those 2,000 genes. Okay, and then using that we are doing the PBMC. Okay, so if you run this, okay, so before that, right, if you like, if PBMC is the object. Okay, so here you will see the something called reductions. Okay, reduction will be empty. Okay, because you have not done any PCA analysis yet. So that's why in this slot of this PBMC object it will be empty. So now let's do the run PCA. Okay, so it will do the PCA analysis, and it has some other parameters as well. So here you can specify how many PCs you want. So by default here we are saying that we want 50 PC, but which we can also do even higher as well. Okay, there is no harm in doing it, but it will take more time. So that's what we need to 50 is more than enough. Okay, so now let's see, it has done the PC, and it finally gives, you see, that information, like for PC1 these are the genes, they positively contribute. Okay, that means when the expression of this gene is increasing, the, let's say, positive side, the PC1 value will be also positive. So they are positively correlated. Or if it is negative value, then PC1 will also negative. So that's what I was saying here, right? The information, which gene is contributing to which PC, which gene's information went to which PC component. That's why this information will give us. Okay, so these are the genes which are positively correlated with the PC1 space, and these are the genes, they are negatively correlated. That means when the value of expression value of these genes increases, the value of the PC1 space, they will decreases. So that way. Okay, and these are the genes, they are correlating towards the second PC. For example, this gene already contributing to PC1, that means this cannot go over here. So the remaining genes, from the remaining genes, which genes are contributing to the PC2 and which genes are contributing to PC3, and like that. Okay, so these are the information. And how many principal component we calculated? We calculated different, by default 50. You can change it to give 60 as well. So now we can just check PBMC dot reductions. Okay, so if initially you see, right, this was empty in the before, and now it has said that total number of cells were this many, and number of dimension was 50, and some other parameter we did not run. Okay, so now we can just you can also check like PBMC PBMC at the rate reductions this slot. Then we have done only PCA. So later on when we do UMAP and those things, like those will also come over here. But now we have done only PCA. That's why we see $PCA. Okay. So if you print this, it will print like that. But yeah, same information you will also get from this as well. PBMC, double bracket, and PCA. So now there are different, we have done the PCA analysis. That means we have got this 50 by 2000. How many? 50 by 2638 matrix we got. Okay. So now if you want to see that, so there is something called visualization loading plot. So here you pass PBMC and say that I want to visualize for first three principal component, and let's run this. I will show you what plot it generates. Okay. So basically we are this plot, what we are seeing, like, for first principal PC1, PC2 and PC3, number 3 principal component, which genes are correlating. Okay, and I'm saying balanced equal to false means any top 5 contributing genes it will display. Okay, so let's do 20. Okay, and for the same safe side I'm just doing 2 principal component, 1 and 2, or let's go with 1 only. So now you see this is the gene, this is the first principal component. These are the genes, they are positively correlated. That's why you see there are two sides, right? Negative and positive. So that means the positive side means they are positively correlated, and negative side means negatively correlated. For example, this MALT1, this gene is negatively correlated. So whenever the value increases, PC1 will decrease. So that way, but positive negatively doesn't matter. But these genes are very, very important. But if you say balanced equal to true, right? So what will happen? It will give 20 genes, okay, 20 features which are positively and which are negatively correlated. So these are the genes which are positively correlated, and these are the genes and which are negatively correlated. So balanced equal to true, you can do that way as well. Okay, and you can say how many dimensions you want. So now 1:2 means now same plot, but we are doing for PC1 and PC2. Okay, even we can go for 1:4. So it will accordingly try to draw the plot. Okay, and you can also tune how many features you want, 20, 30. So here only 20 genes are displayed. If I increase to 30, this will be again more genes will be displayed. But you can also tune other, like the space between the gene symbol in terms of plot, but you get an idea. So this is a very important plot that tells which genes are correlating with which principal components. Okay, perfect. So now this is one kind of plot that you can see, and there are some other parameters are there. So you can go over this one visualization, this parameter as well. So you can change the colors, you can choose which reductions you want. For now PCA, but later on you can use UMAP and TSN as well, and balanced true or false. And you can also divide the plot number of columns like that. So you can try to tune around, play around these other options as well. Okay, but now there are two other plots are there that we will go. The another plot is called dimension plot. So here, so basically this is a very other different kind of plot. So here what we are doing, we are taking two principal component. Okay, so now basically in this my slide is hanged. Okay, so here basically we are taking PC1 versus PC2, and there are 2000 cells are there. We are doing just a scatter plot, right? So that's what we are seeing and how the expression looks like. So here between PC1 and PC2, okay, so all these are the dots, the 2000 dots we see. Okay, and so that's why it shows like we can see that there are three kinds of clusters we can see in the PCA space, and as I said, it maintains the global structure. That means these two cells are very farther from each other in the two cluster. That means the expression profile in real original gene space they are actually different. That's why this preserves the global structure information. And if you don't want to use the legend, so you can remove you can say plus no legend. So this will remove that part. Okay, but legend will be useful. I will tell you how. Okay, so now, but you can see all the cells are same colored. We cannot know what it is, right? So we need to add some metadata to differentiate which cell is what. So now when you say about metadata, so what metadata we have? So in PBMC object, right? So we have, if you remember, like when we did the QC, we have this information, number of count of RNA, number of feature of RNA, and percentage of mitochondrial percentage, like that. Okay, so what you can do also, you can use something called group by. So what we are seeing, group by this cell with respect to some metadata. Okay, so here these all are continuous scales. Okay, so that means they are not category, so like, high mitochondria, low mitochondria, not like, so they are in the continuous scale. So to get, let's say, just to show you an example, so if I am doing PBMC at the rate metadata, okay, and just do a summary function over here. Okay, so you will see that, so this is a numeric, all are, continuous data. That's why minimum equal to 556, 17, and the maximum is 8875. Similarly, percentage of mitochondria, minimum is 0, maximum is 4.994. So that means these are not categorical data. Okay, but still, if you want to group by with that, so you can do same thing, PBMC and PCA reduction, group by this metadata we want. So this name is derived from the metadata associated with each cell. Okay, just to give you some what this metadata is about, if you just do an head PBMC metadata, so each is a one barcode on each cell, and each column is one the metadata associated with the cell. Okay, so if you do this kind of dimension plot, right, so here you will see that you will see different colors here because it's a mitochondrial concentration, right? It's continuous scale. You will see this color. So now what we will do, we'll do one thing. We will I will create this, I will see like anything greater than 2. That means those are high mitochondrial content, and remaining is low mitochondrial content. So that's why I'm introducing another variable here. So if it's a head PBMC at the rate meta.data, so you see that I have something, another class. So that means true means those are high mitochondrial content, and false is equal to they have less than 2. That means those are less mitochondrial content you have. So that way now I have a categorical data. So I can now use this PCA plot and color them with respect to different category. So I just have to now call empty underscore class. Okay, and here I'm giving something called okay, let's see, I was here. So now if I do this one, you will see that I now my PC is divided and color is true color, that mitochondrial true. The true means those are high mitochondrial content, and the red one is are the low mitochondrial content. And since there is no clustering, that means we can say that now the cells are not clustered as per the mitochondrial information. So that means this technical noise, I mean the mitochondrial is not segregating. So whatever the downstream clustering we will get, that will not be because of mitochondrial content, that will be because of the pure biological gene expression. So it's a good indication that you should not get clustering at this stage with respect to this mitochondrial group. Okay, and so now you can sort of group by where all the dots will come in one place. You can also play with split dot by. So here same information, but here the plot will be now split, and this one will be the image from the false, and this will be the image from the true. That means high mitochondrial genes, these are the low mitochondrial genes, and you can see that they are very much similar again. So that shows that means mitochondrial, we have removed the technical noise from with respect to this. Means that means downstream whatever the noise will come, clustering will come, that will be the pure biological signal. Okay, you can also you can play around this alpha parameter to just to the transparency. Sorry for that. You can click here, and you can see that alpha equal to 0.4, and here you can see the dots over there. Okay, so this is one plot. So we saw one plot that is this visualization loading, where gene to PC's correlation we'll see, and here we can between 1 and 2, 2 and 3, how the clustering goes. Okay, and yeah, dimension plot. And also here you can also see which all dimension you want. Let's say you want between PC 1, 2 and 3. Okay, let's try that as well. So you can specify here between 2 and 3. Okay, same plot, but now it will come between 2 and 3. So you see the structure is structure is very different, but again there is no difference with respect to the mitochondrial content, right? Okay, so the third plot what we can do is something called heatmap. This is a very, very important plot. So what you are doing here, heat plot, so here you are giving the object and which PCs you want to visualize. So here we are just focusing on PC1, and here we are telling that take 500 cells to plot. So each line is equal to one cell here, and by default a dimensional heat map function I think it takes around 30 genes. Okay, so with respect to these 30 genes, it tries to cluster. And when I say balanced equal to true, means if you remember here, balanced equal to true means which genes are positively correlated, which genes are negatively correlated. With respect to those, let's say 30, right? 15 positive one and 15 negative one, how the cells are clustered. So now you can see that these genes, for these kind of cells, these cells, they are, let's say yellow means high expression, if they are having very good expression, but this cell, they are having low expression. Same the opposite we see. So that's why how you can see that the high expression of these genes is low in PC1, and that's why we see two kind of clusters. So that means these are very nice information is there, and we can see the clustering between the two cells. Okay, and so here you can also try let's say 15 principal component. Okay, same information. Okay, so now this will be little more information here, but now you can see ideally you should see this kind of contrasting as we move. But this plot helps us next to decide how many principal component to analyze. So in this case you see some difference here, some difference here. That means still we can see some difference, we can see some clustering. Okay, I can see some clustering patch here, and but here little bit degradation is there, signal is not there, right? So here also we cannot see much information, and here it is completely gone. So that means what we can see, whatever the information are there, I think by 8 principal component, okay, cell to cell clustering we can see some distant cluster, but beyond that there is not much information. So in this example, I think 10 principal component is enough to do the downstream analysis. Okay, so that's what the next step is. Next step we need to ask, like how many principal components to choose. So there are two approaches. One is look at this heat map where you see the heterogeneity, and based on that you decide how many principal component. And alternate method, there is something called elbow method is there, so that tells like how many principal component to do. So that we will do. So for that there is something called elbow plot. Okay, I will just click it. So basically what it does, like, so each dot is called one principal component. So what it means, let's say if you just consider 0 to 7 means like is the 7 standard deviation. So huge informations are there. But first principal component has high information, then next principal component little bit less, but third one is little less, fourth one is less, 5 one is less, like that it is coming. But you can see that after 7 and 8 there is an elbow, right? Elbow is like this, right? So there is a band is there, and after that there is not much information. So that means these all are very similar. So that means they are contributing very equal information. So this gives an idea that okay, so now in this analysis 10 can be considered is a good number to go with the downstream analysis. Okay, so this is the principal component analysis that tells. So if you read their documentation, so what they did, like they say that perform linear this one, and Seurat outputs a list of genes with most positive and negative loadings. Okay, and often representing the genes that exhibit either correlation, that means the positive, or anti-correlation, negative, across the single cell dataset. So that's why these are the very important plots. Okay, this kind of plot is an important plot that gives information. And yeah, so next what it says is like how many plots. So there are two approaches. Earlier they were suggesting about some jackstrap based statistical. This will easily give us like okay, consider 20. After that no more information you are passing. Alternate approach is this elbow plot, which will tell that elbow what we saw. And what they are also suggesting here is like telling that let me zoom it a bit. Identifying the true dimensionality could be a challenging. Okay, the first method is more supervised, so that's what you see the heatmap plot. The second is the elbow plot, and third is like whatever the community accepts. So community usually takes 7 to 12 that cutoff for any good data set. So that also you can blindly take it. But another important, this is a very important information that they are saying. For example, let's say you take 10 as the principal component, but what they are saying, like particularly the dendritic cell and NK cells may recognize those genes, those are strongly correlated with 12 and 13. So that means by the 10 or 11th principal component, maximum of the 2000 variable genes, let's say some genes will contribute, but in order to the dendritic and NK cell to pass their information to the PCA, it will come to the PC 12 and 30. Okay, because these are very rare cells, that's what their information will be towards the towards the noise side, not the signal side, because these are the very rare kind of clustering you will see. Okay, for example, that's what they are saying, like these groups are so rare, they are very difficult to distinguish from the background noise. So that's initially PC 1 2 3 4 5 6 up to 10 you get the signal, but towards the end, right, you will get the noise only. So that's why even if you take this, you will not be very it is very difficult to identify these cells. Okay, but there is no harm in taking that. So that's what they are saying, right? We encourage the users to repeat the downstream analysis with different PCs component, like first try with 10 15 and 50, like that you increase and again do your clustering. And but that what they are saying, like as you will observe, the results often do not differ dramatically. So that's what the best is, like you focus on this elbow plot and just choose at what PCs there is no more informations are there, and just you can add one or two extra and then you can go with that. So usually most of the very clean data will have more good by 10 you will be achieving 95% something information. But if it is a very very noisy data, that means even if you go for 50 principal component, you will not see no good contrast or that much. So in that case you can go with higher number, but still that's a very bad data, we would say. Yeah. So that's all about the PCA analysis and how we do. In the next step, basically in the single cell analysis, is the clustering. But now what we got is we got the PC. That means now we have a very clean data and we have a very small data set because we have compressed it. And now the next step is very important, the graph-based clustering. So we need to cluster that into different different cluster. So this is very very important. There are different clustering algorithms are there that I will be talking about, like the KNN, SNN, and what is the resolution. Those are very important concept, because ultimately single cell analysis clustering is important part. So that's why if you don't focus on that, your analysis will be very different. And that's what we will be talking about in the next video. Also UMAP, TSNE, those kind of concepts, like why those are non-linear and how it is different from PCA. Those things we will talk about in the next video. I hope this video was useful, and yeah, I will see you in the next video. Thanks for watching.